Good morning. Please take your seats. I want to welcome you to the presidential lectures. I'm Dr. Matthew Stern, and my co-chair is Dr. Oscar Gershanik. I want to just remind you of what you hear every day, which is to silence your cell phones and that no photography is allowed during the sessions. I should also remind you that faculty disclosure information, which is shown on the rotating slides. This is one of the highlights of the Congress, where we honor two distinguished, long-standing contributors to the field and to the society, and three junior awards that have been chosen by our awards committee for their scientific presentations. The first speaker, it's a great honor to introduce Dr. Günther Deuschl. Dr. Deuschl is professor of neurology at the Christian Albrechts University in Kiel, Germany, and chairman of the Department of Neurology. His team is focused on the pathophysiology and clinical studies of deep brain stimulation for Parkinson's disease and dystonia. He has been particularly interested in tremor, particularly the clinical analysis, epidemiology, genetics, computational analysis, pathophysiology, and neurophysiology of tremors. Professor Deuschl has received many high-ranking awards and honors, like the Max Non and H. Berger Awards. He was the editor of our signature journal, Movement Disorders, from 2004 to 2010. He was president of our society from 2011 to 2013, and is currently president of the European Academy of Neurology. It's a great honor to present Dr. Deuschl to give the Stanley Fahn lecture on the enigma of action tremor in the elderly. Günther.
Presidents, ladies and gentlemen, dear colleagues, it's a great honor to give this Stan Fahn lecture 2015. And if you want to guess how big this honor is, then you just have to look at the relief of my predecessor, which was Joe Jankovic, who gave the lecture last year. Stan, I just wanted to let you know from the very beginning that I will not go as far as Joe did last year, although you look really as amused as I've almost never seen you before. Okay, so let's go to the stuff. I have chosen this title because there are many controversies around essential tremor, but there is one which is really fundamental, and this is this one. There is no consensus so far if essential tremor at young and old age is a cumbersome but mostly benign disease, or if it's a progressive and possibly even life-limiting disease. This issue has a long history, and I will try to just introduce you to that kind of history and discuss a little bit the newer findings in this field. Let's start with this very much respected person. He wrote a paper in 1931 when he was still a junior neurologist at King's, saying that senile tremor constitutes the commonest instance of hypokinesis, although by no means often encountered. Its occurrence in the elderly has been noted from the earliest times by both laymen and physicians. It is indeed one of the traditional stamps of old age. All the others have mostly talked about senile tremor. He was the one who really pinpointed it, and he said senile tremor is its common sites are head, lower jaw, hands, and forearms. Today we would take it the other way around. The amplitude increases as soon as the limb is employed. There is a striking lack of disability. Fine manipulations of the fingers, however, soon become impaired. And already in these days, he said, possibly senile tremor represents a delayed form of the idiopathic family tremor, suggesting that the frontier between essential and the senile form is ill-defined. Well, we could say the same thing today. There's no change. Well, in these days, there were two positions held. One was held by this person, Professor Meiner, who was a Russian neurologist coming from Latvia. And he came to the conclusion that essential tremor has something like a status macrobioticus multiparus, which means that it's a condition characterized by tremor, longevity, and fertility. So for him, it was an advantage to have an essential tremor. And in 1949, Critchley summarized the situation as it was standing in these days. In the literature, there are accordingly two contrary viewpoints, namely that essential tremor is a sort of macrobiotic manifestation, and conversely that it is a degenerative disorder associated with the neuropathic family taint. Despite the size of the literature, there is still an inadequate documentation. True until today. Just a remark. Degenerative in these days meant something different than today. In these days, it meant that it's not only this symptom, but it has also other neuropsychiatric connotations. Other neuropsychiatric conditions occur with it. So this is the long-standing controversy about which we are talking. We have tremor as a sign of aging, and ET comes with neurodegeneration that has been put forward in the 19th century already, and then later in the 20th century. And just now, there is a revival of this hypothesis. And on the other hand, there was a group of researchers and clinicians which believe that ET comes with unaffected or even longer life expectancy with mostly limited morbidity. And that has been put forward by Pellner. That's the first one where I found this like this. Joe Jankovic has written a paper in 1995, which I would like to particularly mention. He has done a study in these days, which is probably not really, is not compatible with modern methods, but he just looked at the age at death of the parents of patients with essential tremor and compared that to a control group. And thereby he found that the parents of patients with essential tremor were on average 8 years older. Okay, modern findings in favor of neurodegeneration. Pathology is certainly the first point that comes to your mind when it comes to neurodegeneration. There have been 19 cases since the 30s, and in 2004, the knowledge about the neuropathology of essential tremor was just doubled. But all these 40 cases found inconsistent pathology, and it was not until Elan Lewis published a number of papers, and overall he claimed that there are two types of pathology. One is ET with Lewy bodies, another is ET without Lewy bodies, but they have a Purkinje cell loss and torpedoes. Holy Schill had 24 patients, that's the group in Arizona, and they found inconsistent pathology and could not confirm the findings of Elan. And then there was Alex Rajput, another group that is doing this kind of research since decades, and they found that Purkinje cell loss is age-dependent and not specific for ET. So that's where the situation stands, and the Movement Disorder Society and also the NIH are very much interested in this, and we recently had a meeting and we agreed that these three groups have to go together and sort this out, whether there is pathology or not. But I wanted to put forward an idea which I simply cannot throw away. After 50 years of a disease, people die mostly from other reasons. And then neuropathologists look at these brains, and then they have problems to see the same features. This is just difficult to understand for a simple clinician. Really, the evidence in favor of pathological neurodegeneration is probably quite limited. Can genetics identify a genetic abnormality related to neurodegeneration? Well, there are, as you know, 3 loci in linkage studies, but no gene has been found so far. There are 2 risk genes in GWAS studies that show up, but and they may be related to neurodegeneration, but as you know, it's a very borderline finding. And there's one family with a FUS gene mutation, and the FUS gene, as you know, is involved in ALS, so that's a definite probable hint towards a neurodegenerative process. But this genetic finding has not been confirmed in other cohorts. So the best evidence for neurodegeneration, in my view, is the clinical evidence, and that's epidemiological evidence. There are 2 large epidemiological studies in New York and in Spain, and prevalent dementia is increased in ET in the New York study. This is also true for the Spanish study, but there it's only for those cases above older than 65 years. And also incident dementia is increased in ET with onset beyond 65 years. But keep in mind, there is, and there's even a higher mortality in ET, but keep in mind that there is still the Joe's finding from 1995. So the question is, can age at onset help to discriminate subgroups of tremors? Well, if you look at the prevalence of essential tremor, then you see that starting from the age of 40, the fifth decade, the prevalence is increasing very much until almost 10% in subjects in cohorts with an age above 90. We have looked at this issue in more detail with the Danish twin registry in Odense, which is relatively close to Kiel. This was already in 2001, and in these days they had 2,327 subjects in the study. They were all older than 70 years. They were all single twins or intact twin pairs. The subjects were assessed in 2001 with a very extended interview and some aging tests, so hand grip, neuropsychological standard tests, activities of daily living, medication, and morbidity. Tremor was assessed with the Archimedes spiral. This group has special features. First of all, the old age at inclusion, older than 70 years. We have a long observation period between 2001 and 2014. Most of the patients are already dead in 2014, and hence the event rate of mortality is very high. It is a unique cohort because we have the connection between this cohort and national databases of Denmark, mortality data, healthcare system data. That's all possible in Denmark, and therefore the databases there are so important. But we have a limitation. The personal assessment was only done in a subgroup of 12%. But even that, Delia was doing that in 2002, and even that took 9 months for her. Well, this is a very important contribution of Stan. He wrote the most important tremor rating scale in '93, and he published it with Conchita Marin and with this particular person here. You see also the other, so maybe that was around that time, and maybe they were just celebrating a paper in these days. You see here Stan, and you see here David in a very characteristic position. We all know this from these days, and it's one of the highly cited of your papers, Stan, actually 584 quotations. But we didn't have this in all these 2,300 patients. We looked at tremor severity by looking at spirals. And this procedure is used by many groups, and it has shown validity, test, retest, reliability. it is a very good test, and we have data of this test comparing that with the Fahn score. The Fahn score is a personal assessment, as I said, and when you look at the relation between the tremor severity according to the Bain score and the Fahn score, you get this. So this is almost a perfect logarithmic relation between them, and therefore we believe that we do have a surrogate parameter with these tremor score for tremor. And that enables us to look into the data from this particular cohort. Oh no, before I go into the details, I have to mention another point. The question is, what is an abnormal spiral? If you look here, where does tremor really begin? Is it here or is it there? So we have made the following decision. We have just said grade 3 means that most of the spirals are with a rhythmic activity, as you can see here, but there are some which do not have clear rhythmic activity, while at grade 4 all of them do have it, and that's how we rate them. So grade 4 is, in our view, a manifest tremor, while grade 3 is borderline. So how do the spiral score distribute in this population? Well, we have about a quarter that has a pathologic tremor, that has a symptomatic tremor. The others have less intense tremor severity, 3, or they have no tremor. And this enables just to compare how these people here, how they differ with respect to some aspects that we have in the database. First of all, how is the mean spiral score in the different age groups? And what you can see is that the different age groups, there's a clear increase between 70 and 90. You can look at the prevalence of symptomatic tremors in subjects with an age above 70, those which have a spiral score of 4 and greater. And you see that this is true for 20% of the patients, of the subjects that are not patients, between 70 and 74, and it's almost 90%, almost 50% in those above 90. So it's a very frequent condition. And then we can look at the hazard ratio for mortality. And that is also heavily increasing with age, meaning that when you compare it with the reference population, which is 0 to 2 spiral score, then at a spiral score of 5 to 7, you have a hazard ratio of 1.84, which means that the likelihood to die at any age between 70 and 90 is 84% higher than the control population. This is probably the most important slide of my presentation, because it clearly shows that the survival curves are depending on the spiral score, on the tremor severity as shown here. So this is no tremor, this is tremor 3, this is 4, and this is 5 to 7. How can we measure aging? So far we have measured tremor and we have measured mortality, but we want to know if tremor is also related to aging and not only to mortality. Well, you can look at epidemiologically established aging parameters. One of them is grip strength. Just to note, aging does not begin at the age of 60 or so. It begins pretty early, ladies and gentlemen. So even at 35, your grip strength is decreasing. Also, your cognitive composite and mobility score is decreasing. These are very strong data from large groups that have been investigated with these parameters. And now we can use these parameters to look at our tremor group. And this is what I'm going to do in a second. So, what did we find? Well, we found that grip strength, for example, is decreased depending on the tremor score. So 75 kilopond less, 0.9 kilopond less for tremor severity 4, and for the group 5 to 7, it's almost 3 kilopond less than the control population. The same applies for the cognitive composite and for the ADL score. If we know that it is related to aging parameters, if that tremor is related to aging parameters, then we certainly want to know, is it explained by these aging parameters? And we have to look for the hazard ratio, but we have to control for all these parameters. And what you can see is when you control for age, sex, medication, grip strength, ADL, and cognitive functioning, there is still a significant hazard ratio of 1.25, meaning that the likelihood to die is 25% higher for a tremor subject, which is spiral core 4 or above. So to get this very clear until here, tremor in the elderly is a risk factor for mortality that is strongly related to aging parameters. Even when we control for classical aging factors, the mortality risk of tremor is increased by 25% at any age above 70. That's the conclusion from this study. Tremor is therefore not an innocent bystander of bad health. It has its own effect on mortality, and it is a new risk factor for mortality. And just clinically speaking, late-onset tremor is a senile tremor. Senile tremor is probably politically no longer correct, and therefore we propose to name this aging-related tremor. What about classical essential tremor? Does it fit with these features? Well, we have these 276 subjects which Delia has visited, and 36 of them were definite ET, 69 probable ET, and we can look certainly at the parameters that we have looked in the larger cohort. And what you see here is that the definite ETs are not, do not have a similarly increased hazard ratio for mortality. The hazard ratio is lowered. It is lower. And it's also lower for the possible and probable ETs, while it's the same for the other tremors as for the senile tremors and for the age-related tremors. But as you can see here, findings are not significant, and therefore they are not much of a value. But there are significant findings regarding the aging parameters. They are not worse than the control population. They are better. So they have a stronger grip strength, they have a better cognitive score, and they have a higher ADL score, and that is significant here. So this part of our study is clearly underpowered, but so far there's no evidence that mortality is increased in classical early onset ET. So probably these two conditions differ. So we asked ourselves if we can separate early onset from aging-related tremor clinically, certainly a very obvious question. We looked at the ET cohort in Kiel, and we found almost 1,000 patients with definite or probable ET that we have collected over the past 15 years. And these people were all involved in this study. The age at onset in the Kiel cohort is, you have here a peak at young age and a peak at old age, so we just can dichotomize the group and can look at those, at the clinical differences of early and of late onset tremors. And what you see, first of all, male-female ratio is the same in both conditions. Men are more often suffering from tremor than female. The tremor severity is not significantly different in the two groups. The topography in the two groups is not significantly different, but certainly the mean age and onsets and the disease duration just by definition. Are there further clinical differences? Yes, there are. Alcohol responsiveness is higher in the early onset group, and family history is higher in the early onset group. And the tremor progression is faster, much faster in the late onset tremor group. But all these features don't allow you to say, when you have a particular patient, this is an ET patient or an ART patient. So there is no clinical way to look at this. We have been looking at electrophysiological differences between early onset and late onset tremor. And just what kind of electrophysiological differences? Well, there's a tremor network, as many of you know. There are places in the brain that are activated related to peripheral tremor. And the way you look at this is that you put patients either in an MEG or high-density EEG, or even in an MRI scanner, and then you look at the related activity in the brain to the peripheral tremor. And then you find these activations, these cortical activations in the primary motor cortex and the frontal cortex, mostly in the thalamus and in the contralateral cerebellum. This has been nicely shown for Parkinson tremor. It has been shown for Wilson's tremor. For essential tremor, here you have an additional source in the brainstem in all of them, and we have shown this also for orthostatic tremor. And you see that sometimes you have a very precise location of this activation. We have been comparing early onset and late onset tremor in two groups of patients. These were the people who were involved in this study. We have used high density EEG, and tremor was used as tremor activity was used as the reference. And here you see the two groups, which by itself is quite interesting. These were just groups, just patients that we could get hold of, which fulfilled either the criterion of early or late onset, and which were matched for age. And you see that in this group, certainly age at onset differs very much, and disease duration just by definition. The tremor severity is the same, and also the distribution is the same, but already in this small group you see a difference of muscle strength, and you see a difference of the composite score in these 10 versus 10 patients. So probably in the future we will diagnose ART by making neuropsychological tests. The pattern that we found with this kind of assessment was the classical pattern of ET. You can see this here. Motor cortex, prefrontal cortex, thalamus, cerebellum, and brainstem were all activated, and we can also look at the directed coherence. This goes too far at this moment, and it's not necessary, but it was like we know it from many other cases with essential tremor, and this is the pattern of aging-related tremor. So we don't have a source in the brainstem and in the cerebellum in this particular group. And we did a second group, certainly, and again, we didn't find it. It was controlled for all the parameters, tremor severity and EMG, amplitude, and whatever. We couldn't find a source below the thalamus. So that may be in the future another way to look at it, because under these circumstances you have to consider other ways how this tremor is caused, and we may want to look at this. So this is the situation. You have a corticothalamic origin of this aging-related tremor, while it is different for the classical early onset ET. So let me summarize. We have found that late onset tremor is increasing with age and aging parameters, but a separate risk factor for mortality. So we propose to call this an aging-related tremor. Motor features of ART are indistinguishable from early onset ET, but aging parameters like cognition and grip force may help. Perhaps we have to diagnose this subgroup in the future just by looking at cognition, by measuring grip force, and having a reassessment after 2 years. The underlying tremor network seems to differ for these two tremor forms. And finally, early onset ET and ART are probably different conditions. And if that is the case, we would have solved the enigma of action tremor in the elderly, because then we have the ART as one form of tremor and the early onset essential tremor as the other. Thank you very much.
We're now going to move on to the junior award lecturers. The first lecture is by Mashiao Raman from Genji, Korea, who's going to talk about the mitochondrial serine protease HTRA2 degrades disease-associated prion protein. Mashiar, is he here? Where did he go? He was here. Oh, okay. Let's move on then. Are you guys ready? Yeah, okay.
No, wait. Don't rush. Well, great pleasure. I would like to announce the next young awardee, which is Karen Maurer from NIH, and she will talk about mechanisms underlying impaired self-agency in functional movement disorders, a resting state F functional MRI study. Dr. Maurer?
I think so. Okay. All right. Good morning, and thank you for this great honor, and thank you for giving me the opportunity to speak about our work on the mechanism of impaired self-agency in patients with functional movement disorders. So what is self-agency? Self-agency refers to the subjective perception that one is initiating, executing, and controlling one's own actions. So put quite simply, this self-agency allows us to experience that we are the cause of our own actions. How is this sense of self-agency generated? One influential model for the mechanism of self-agency is that of the comparator model. In this model, there is a constant monitoring of whether motors and actions' predicted consequences equal or match up with the perceived consequences. So motor intention generates a prediction of motor consequences. This is then compared with the perceived proprioceptive consequences that occur after an action has been performed. The comparison of predicted and perceived occurs at a brain region termed the comparator, also known as the mismatch detector. When perceived motor feed forward matches with, when predicted motor feed forward matches with perceived sensory feedback, a sense of self-agency is generated. Impaired self-agency is one of the key features of patients with functional movement disorders. So, as is evident on interview, patients with functional movement disorders feel that their abnormal movements are involuntary, despite the fact that we know that physiologically these movements utilize voluntary motor pathways. This impaired self-agency is also evident in various implicit measures of self-agency. One of these is termed action-effect binding. So in this paradigm, an action, for example, touching a domino, that is followed by an effect, for example, the dominoes falling down. In healthy subjects, there's a temporal binding of this action and effect such that the healthy subjects perceive their action as occurring later and the effect as occurring earlier than they do in reality. In patients with functional movement disorders, this action-effect binding is impaired. Patients with functional movement disorders also exhibit impaired sensory attenuation. This is a phenomenon whereby in healthy subjects, sensory intensity of a self-generated movement is impaired, is decreased. And this decrease is impaired in patients with functional movement disorders. So what makes and what distinguishes involuntary versus voluntary movement. So Valerie Voon and colleagues in the Hallett lab sought to answer this question several years ago in using a within subject fMRI block design task. In this experiment, they compared functional involuntary tremor with voluntary mimic tremor in 8 patients with functional tremor. And intriguingly, they found that in the case of the functional tremor, there was decreased activity of the right temporo-parietal junction. So what is the implication of this decreased activity in the right TPJ? So work from numerous studies at this point has implicated the right TPJ as playing an important role in self-agency, notably as playing the role of mismatch detector. So it's the thought to be the right TPJ where predicted motor feed forward is compared to perceived sensory feedback. Other work from the Hallett lab performed by Nahab and colleagues has implicated the right temporo-parietal junction as being involved in the leading network of self-agency. So in this work, Nahab and colleagues performed a virtual reality experiment in the scanner, and they identified 2 networks that seem to play an important role in generating a sense of self-agency. They term these networks the leading network and the lagging network. The leading network was thought to play a role in mismatch detection and includes the right TPJ. The lagging network is thought to play a role in bringing this information into conscious awareness. So we were interested in further investigating the mechanism underlying impaired self-agency in patients with functional movement disorders using neuroimaging tools. Specifically, we used resting state functional MRI in order to examine this self-agency network. So resting state functional MRI is a method for evaluating functional neural networks occurring in the absence of any explicit task. Brain activity, as we're all aware, occurs even when we're not explicitly performing a task. In resting state functional MRI, functional neural networks are identified by correlations of spontaneous signal fluctuations in blood oxygen level, which are referred to as the bold signal. In our study, we performed resting state functional neuroimaging on a relatively large sample of 35 patients with clinically definite functional movement disorders and an equal number of age and sex-matched healthy volunteers. We wish to investigate the differences in the neural networks underlying self-agency, and we utilize the right temporo-parietal junction as our seed in comparing these 2 groups. Study participants were well matched in terms of age and sex. All participants underwent extensive psychological assessment. As expected, patients with functional movement disorders scored significantly higher on scores of anxiety and depression. Clinically, patients tended towards the more chronic illness, with an average symptom duration of about 5 years. They reported a variety of abnormal movements, with tremor being the most common abnormal movement. Structural and functional imaging was obtained using a 3 Tesla scanner. Anatomical images were obtained using T1 weighted MP RAGE sequence, and functional images were obtained using T2 weighted multi-echo planar sequence. During the acquisition of functional imaging, the patients were instructed to close their eyes and try not to think of anything in particular. Data preprocessing and analysis was performed using the AFNI software. Group-wise comparison was performed using the AFNI tool group Instacore. We compared groups by using between subject to sample t-tests and used age, sex, depression score, and childhood trauma score as covariates. So we were interested in in looking at resting state functional connectivity from the right temporo-parietal junction and comparing this connectivity in controls and patients. As our seed, we used a coordinate that was derived from a meta-analysis performed by Desity and Lamb, in which they looked at the right TPJ and its role in self-agency and they looked at this across 15 different neuroimaging studies. Shown on this slide is the maps of the seed-based correlation analysis using the right TPJ as our seed and the different maps for the controls and patients. So we were interested, as I indicated, in how these were different. So shown on this slide is the map of on the right-hand part of the slide is a map of the group differences in resting state functional connectivity from the right TPJ in controls and patients with functional movement disorders. Looking at some of the regions that we picked up in greater detail, we saw impaired resting state functional connectivity between the right temporo-parietal junction and several sensory motor regions. So specifically, we saw decreased functional connectivity between the right TPJ and bilateral sensory motor cortex and bilateral cerebellum. So we hypothesized that this reflects impaired motor feed forward and impaired sensory feedback to the site of the brain's mismatch detector. We also saw decreased functional connectivity in patients between the right TPJ and the right insula and left supplementary motor area. These areas are, both the insula and the SMA are implicated in intentional binding, which is a process whereby motor intention is paired with sensory feedback. We hypothesized that this impairment in functional connectivity between the right TPJ and these areas of sensory motor integration may play a role in the impaired sense of self-agency in patients with functional movement disorders. So shown on this slide is the list of all of the areas that we found that to have diminished functional connectivity in patients as compared to healthy volunteers. And intriguingly, a large majority of the regions that we found have already been implicated in a sense of self-agency. Those areas that I have highlighted in red, which showed decreased functional connectivity to the right TPJ in our patient group, have also been notably identified as playing a role in the leading network of self-agency that I had described to you earlier in the talk. So to conclude, our work provides further evidence supporting abnormalities in functional connectivity in patients with functional movement disorders. Furthermore, these abnormalities are present during the resting state and not merely during performance of task. These abnormalities are independent of other comorbidities as assessed by given that we included these as covariates in our analysis. And these abnormalities implicate impaired sensory motor integration at the site of the brain's mismatch detector. So our work supports a model whereby there is impaired motor feed forward, impaired sensory feedback, and impaired sensory motor integration to the site of, to the right TPJ, the mismatch detector, and we propose that this contributes to the impaired sense of self-agency in patients with functional movement disorders. So I'd like to thank everyone for their attention, and also this work was accomplished with the help of many. I wanted to thank first and foremost my PI, Mark Hallett, for guidance and for discussions. Sylvina Horvitz, the MRI guru in our lab, for assistance at all steps along the way. Catherine Lefevre, a former fellow in the lab who initiated the study and performed the initial MRI acquisition and patient recruitment. All of the patients and volunteers. And I wanted to again thank the organizers for this great honor and to speak here today.
The next speaker is Bettina Balin from the UCL Institute of Neurology in London. Her topic is antibody spectrum in stiff person syndrome and related disorders.
Hello. Good morning. Dear colleagues, before I start talking about antibodies in stiff person syndrome and related disorders, I would like to thank the awarding committee for having me here and the chance to present our work. And even though I have the honor to stand here today, this work was really teamwork, and it would not have been possible without the contribution of each single of my colleagues over here. And first of all, my mentor, Professor Mink, who passed on to me his enthusiasm for stiff person syndrome. By Merchant Waldman, who encountered patients with a rare and enigmatic disorder causing stiffness and spasms, and leading to this typical, also hyperlordotic posture. And subsequently, it became clear that the clinical spectrum is much broader, also involving focal variants like the stiff limb syndrome, or variants with a more widespread involvement and a potentially lethal disease course, like progressive encephalomyelitis with rigidity and myoclonus, PERM. And also there are some patients which have additional neurological signs which differentiate them from classical stiff person syndrome, but do not feature the aggressive course as we know it from the initial descriptions of PERM. So for example, this patient over here, which we call a variant of PERM. All of these I will summarize in this talk under SPSD, stiff person spectrum disorders. And the broadening clinical phenotype was paralleled by increasing recognition of different antibodies, mainly targeting proteins of inhibitory GABAergic or glycinergic synapses. So first of all, GAT antibodies, then amphiphysin antibodies. Then Jeffrin antibodies have been so far described only in one patient who had a paraneoplastic stiff person syndrome with mediastinal cancer. And GABA receptor-associated protein antibodies, GABA-RAP, were described by one group in patients who also harbored GAT antibodies. Then, in 2008, Angela Vincent's group described for the first time glycine receptor antibodies in a patient of PERM, and subsequently we learned that these antibodies are not unfrequent. And in the last year only, 2 new antibodies have been described in SPSD. So Professor Dalmau's group has found GABA-A receptor antibodies in 4 patients with SPSD, partly co-occurring with GAT antibodies. Usually, GABA-A receptor antibodies associate with an encephalitis with refractory epilepsy. And we have found TPPX antibodies in a variant of PERM. And these antibodies have also been initially described in an encephalitis, a protected encephalitis with prodromal diarrhea. So why is this so interesting and exciting? Current concepts in neuroimmunology assign pathogenic relevance to neuronal surface antibodies because the antibodies can they target proteins which are on the neuronal surface and therefore accessible for the antibodies in vivo. And these antibodies, or the group of patients which harbor these antibodies, seem to have also a better treatment response. And this stands in stark contrast to the classical onconeuronal antibodies like HU, YO, RI, which are rather a biomarker for paraneoplastic process. And in a somewhat intermediate position are the intracellular synaptic antibodies like GAT and amphiphysine, where we don't really know if they can have some pathogenic relevance or if they're just a biomarker. So with this in mind, we wanted to explore the spectrum of the new antibodies in stiff person syndrome. So we looked for antibodies against GAT, which is stands for glutamic acid decarboxylase, the rate-limiting enzyme in the synthesis of the main inhibitory neurotransmitter GABA, amphiphysine, which is important for the endocytosis. I talked before a little bit about glycine receptor and the GABA-A receptors, and Geoffrin is important for the glycine receptor anchoring. And all these are basically rather mechanisms which impair inhibitory, the inhibition in GABAergic or glycinergic synapses, whereas DPPX holds a somewhat extra position in here. DPPX is a regulatory subunit of voltage-gated potassium channels, the KV.4 channels, and its loss of function results in neuronal hyperexcitability. And while we are thinking of doing this, another thought occurred to us. So if you look at SPSD, it shares some clinical hallmark features with hereditary hyperplexia. Patients have an exaggerated startle response. They may have stiffness and spasms. And therefore, we wondered if proteins, which are mutated gene products in hereditary forms of hyperplexia, might lend themselves as good targets for antibodies. If you think of it, glycine receptor antibodies targeted the alpha-1 subunit of the glycine receptor, and this is actually just a gene product which is in one of the affected in one of the major forms of hereditary hyperplexia. So there are other genes which are involved in hereditary hyperplexia. For example, genes encoding for the glycine transporter 2, or genes encoding for the beta subunit of the glycine receptor. And I have mentioned Geoffrin before. Colibistin is also important for anchoring of the glycine receptor. And also, not from the genetic side, but because GABAergic neurotransmission seems to be the linchpin mechanism in SPSD, we were also interested in looking in GABA-B receptor antibodies. So what did we had a twofold approach, therefore, looking for the prevalence of known antibodies, like particularly the new ones, GABA-A receptor, DPPX, the glycine receptor antibodies, the classical one targeting the alpha subunit, and GAT amplifying Geoffrin. And on the other hand, we wanted to explore novel antigens, like the glycine receptor beta subunit or the glycine transporter, colibistin, or antibodies which have been so far never described in SPSD, like GABA-B receptor. antibodies. So we looked in the serum more than in the CSF in a big cohort of SPSD in 73 patients with different forms, SPS, SLS, PERM, and in 5 patients with acquired hyperplexia. I reviewed also the clinical and paraclinical data, including antibody test results from our lab or the lab of Professor Vincent in Oxford, and I was also looking at the CSF data and MRI. And we used indirect immunofluorescence on frozen tissue sections and transfected tax cells and ELISA. So what did we find? This pie chart shows the whole antibody spectrum in SPSD. So first of all, we had to realize that there's a quite significant amount of seronegative patients, 10%. Now if we look at the seropositive cases only, it becomes clear that gut antibodies are still the most frequently found antibody, and the clinical spectrum spans from SPS to PERM and SLS. And they occur either alone or in conjunction with other antibodies. Amphiphysin antibodies associated strongly with paraneoplastic stiff person syndrome and occurred either alone or together with other antibodies at low titers. The glycine receptor antibodies were the second most frequent antibody in SPSD, and it was found in stiff person patients and PERM, occurring again either alone or with other antibodies. DPPX antibodies are overall not frequent in SPSD, more so given that we have included in our study previously reported patients. And they occurred again either alone or with glycine receptor antibodies at lower titers. So another remarkable thing I have pointed out while I was talking was the co-occurrence of antibodies in 12% of the seropositive cases. In the acquired hyperplexia group, we found one patient harboring glycine receptor antibodies and Jeffrin antibodies, and this patient had an idiopathic brainstem encephalitis, meaning there was no paraneoplastic etiology like in the firstly described patient. Another patient harbored CA antibodies. This is not an antibody we tested in our panel. This was an antibody which was found in this patient before, and I noticed it in reviewing the data. CA antibodies target Purkinje cells. And the majority of the patients, the remaining 3, were seronegative in this group. Now, you might have spotted before on the pie chart that I had also some antibody from the second novel antigen approach group, which was the glycine transporter 2. So we found 2 cases with glycine transporter 2 antibodies co-occurring with GAT antibodies. However, unfortunately, I guess this is real life, data were only available for one patient. And the phenotype was somewhat indistinct, I would say. So it was a man who's now 61 years old, who started having first symptoms age 50, with insidious onset of leg stiffness. Then he developed a characteristic fear of walking unaided. He had then an diplopia and gait ataxia. And investigations-wise, the CSF showed bit raised protein and an impaired blood CSF barrier dysfunction, but otherwise nothing remarkable, and the MRI was normal. His treatment response to steroids was initially good and then poor. Glycine transporter 2 is located presynaptically in glycinergic axons in the brainstem or the spinal cord, and glycine transporter 2 holds a critical role in maintaining a high presynaptic pool of glycine. And its mutations are the second major cause of hereditary hyperplexia. So before I summarize our results, let me also tell you about the limitations of this study. So in terms of study design, this was a retrospective study with the usual associated shortcomings. However, if you would like at a clinically defined cohort of a very rare disease, I think this is probably the only way how to do it. Our approach, this hypothesis-driven approach, thinking of which targets might be affected by antigens, trying to learn from genetic forms, is also a bit unusual and differs from the more common approach with identifying similar immunostaining, immunoprecipitation, and mass spectroscopy. However, for example, the glycine transporter 2 gene was also found by hypothesis-driven approach. So the authors actually were thinking which targets could be involved in this disease and found mutations. Methodologically, we used mainly serum, and ideally, of course, you would like to use paired serum and CSF samples. However, stored CSF samples are not available in big amounts. And what we can't really say is what is the influence of storage and freezing and thawing on the antibodies. Robust data about this exists for aquaporin 4 antibodies, which are again quite robust to storage, thawing, and these, but we can't for sure say that this applies to all antibodies. So let me summarize. In terms of the prevalence of the known antibodies, the GAT, the classical glycine receptor antibody, and amphiphysine account for the majority of the seropositive cases. In our intent to find novel antibodies, we found glycine transporter 2 antibodies, but none of the other hypothetical antigens was binding anything. The lessons for clinical routine, which I take from this, is one antibody can have or can be found in many different phenotypes, and one phenotype can have many different underlying antibodies, and multiple antibodies occurred in a significant number of patients, so this would suggest that one should rather do panel testing. And lastly, for the future, well, we will have to do further studies to look into the clinical significance of glycine transporter 2 antibodies and into the pathophysiological mechanisms. We'll also have to think of the significance of lower titer amphiphysine and DPPX antibodies, as quickly mentioned before. And a high proportion of seronegative cases highlights that further efforts are needed to identify the full spectrum of antibodies in SPSD. Thank you.
Now moving on to the other presidential awardee, it is a great pleasure for me to introduce Professor José Obeso, not only because of his distinguished career as an expert in movement disorders, but he is especially a good friend of mine. We started together in the field of movement disorders many years ago, and he was a good friend then, and he's still a good friend now. Dr. Obeso, who is a graduate from the University of Navarra, had his main instance of approaching the field of movement disorders when he had the chance between 1980 and 1982 to work as a researcher under the mentorship of Professor David Marsden, one of the founders of our society, in his lab in London. Dr. Obeso is currently director of the CINAC at the Hospital Puerta del Sur in Móstoles, Madrid, Spain, and full professor of neurology in the CEU San Pablo University in Madrid. He has made significant contributions to the field in the areas of pathophysiology of the basal ganglia and surgical treatment of Parkinson's disease. He's also the chief editor of a signature journal, Movement Disorders, the leading publication in the field of Parkinson's disease and movement disorders. He's also an honorary member and invited professor of several organizations and institutions, such as the Colombian Neurological Association, the French Neurological Society, the Bolivian Neurological Society, and he's a professor of the University of La Habana in Cuba. Dr. Obeso, he's going to talk about basal ganglia and pathophysiology, puzzles, paradoxes, and clues.
Thank you very much, Oscar. Good morning. It's a big honor and pleasure to have been appointed to give this lecture today. Let me see how I The lecture will be dealing with, as it is customary, with the name of the lecturer, Professor Marsden. In this case, I'm going to take the liberty of being a little bit in more detail, since Marsden was my mentor, my professor, my friend. Also because the tight work we did together on the pathophysiological models of the basal ganglia, I'm going to quickly revise some of these aspects, as well as the classic paradoxes derived from the model, to allow more time to my current field of interest, coming from how findings in relation with basal ganglia pathophysiology may give way to some clues to understand dopamine cell vulnerability in the nervous system. Now, Marsden started his academic career with Professor David in St. Thomas Medical School with his master's degree on the substantia nigra in the cat. And this is how he apparently became involved, passionate by the basal ganglia. He did a quick clinical training period in Queen Square, and then at the premature age of 34, was appointed professor and consultant of neurology in the Institute of Psychiatry. This was the first chair of neurology across the river, and ever since, south the river in London became a trench in competition with the mecca of neurology north in Queen Square. The evolution of Marsden academic life was really rocketing. He was elected the youngest member of the Royal Society. He was among the 10 top authors in the 80s, and even after his death many years ago, he has this impressive ACE index among the 10 tops for all neurology and neuroscientists nowadays. But his real interest was in understanding mechanism, and Professor Pat Merton from Cambridge, who was a very in-depth and proper physiology, was his mentor from this point of view. They embarked to demonstrate that this stress reflex, the so-called low latency stress reflex, was using a cortical mechanism that caused tremendous polemic in the 70s and 80s, and they published many papers, famous by the Marsden, Merton, and Morton authorship, giving all sorts of indications to prove that indeed this is a cortical mechanism. In London, he worked in the Institute of Psychiatry. This was his office. He spent many hours. It was difficult to arrive before or leave after Marsden in those years. The real good thing, if you stay there for long enough, is that at the end of the day, we could come out here, turn the right, and we'd go to this pub or this pub, and here is where the real good ideas on the best papers would keep written down here, and then off you go for the weekend to come back on Monday with a paper. In the 90s, when Marsden was at the peak of his career, this classic, what is now called classic model of the basal ganglia, was developed. Essentially, it shows that the striatal cells project in the direct pathway to the GPI and indirectly through the GPE, subthalamic nucleus, to the GPI. This is inhibitory and this is excitatory because of a double inhibition, and therefore it is supposed that either both pathways maintain an equilibrium of control, one of facilitation, one of inhibition. Alan Crossman in Manchester played a fundamental role in developing and studying the subthalamic nucleus, but of course it were the paper from Alvey, Penny and Young, Malon DeLong, that had such a massive, major impact in the community. Essentially, the model just state that pulses in the output facilitate movement by disinhibition, discharges will reduce movement by increasing excitability on the pallidothalamic projection, and therefore from here, Anne Young and Penny and DeLong easily deduced that excessive activity could lead to Parkinsonism, and movement inhibition, and reduced activity could lead to involuntary movement of chorea. And this is the essence of the model. The model was supported by enormous amount of data in the 90s. The MPTP monkey allowed to show that indeed there is hyperactivity in the subthalamic nucleus, and GPI, recording neuronal activity. The GAD, the GABA enzyme, is increased in this nucleus and is reduced in monkeys after a subthalamic nucleus lesion. In patients, there is hypoactivity of the cortical premotor area and MSA before lesion. After palidotomy, these areas became more active, indicating that their their excitability has been restored by a local action down in the basal ganglia to stop this excessive inhibition. And of course, we all know the tremendous impact of motor improvement with surgery. More modernly, more recently, by optogenetic, it's been shown that indeed by selective activation of neuronal groups like these D2 neurons, medium spiny neurons in the indirect circuit in the striatum, can actually, as predicted, if this is a stopping inhibitory system, you will see that when these neurons are activated by the laser coming here, the mice stop, the laser goes, the mice move again, the laser comes, and the mice will stop again, showing that indeed the indirect circuit is very capable of stopping movement by activating that pathway. So with this in mind, one could somehow concede and conclude that the model fit with the notion that the motor circuit is normally involved in selection, facilitation of movement, and as a result, when this is faulting, bradykinesia is secondary to abnormal energization of neuronal activity in the motor circuit, a notion that Mark Hallett put forward in the early 80s. Remarkably enough, in my mind, and for the now almost 20 years the model has been around, there has been little attention to the fact that the two other major clinical features of Parkinson's disease, rigidity and tremor, have not really been understood and scrutinized from the same point of view. And if one thinks about rigidity, which is obviously a positive, a release sign in Jacksonian terms, you see all these changes in the Parkinsonian brain. There is increased motor cortical excitability, there is increased glucose metabolism by FDG in the motor cortex. The long latency stroke reflex, as shown before by Marsden himself, is increased, and in the MPTP monkey, there is increased neuronal cortical response. So this suggests actually that the Parkinsonian brain, and even for two tight motor features like bradykinesia and rigidity, they don't actually go together. This could suggest that there are two different mechanisms, and at one hand, we could have this premotor circuit going through, which is the one to the premotor areas and presupplementary area, which is hypoactive, while the primary motor cortex, basal ganglia, back to the primary motor cortex, also with a cerebellar influence, is actually hyperactive. And that may be, just may be, part of what is going on to explain rigidity. Similarly, tremor poses a tremendous puzzle here, because tremoric activity is present in many regions of the brain, in the basal ganglia, out of the basal ganglia, in VIM, which is a cerebellar receiving area projecting to the motor cortex, etc. So how these two circuits, the one where the dopamine deficit originating the tremor is somehow driving and recruiting this other abnormal circuit, which should be normal from the cerebello-thalamocortical pathway. This is really not understood, and is really a sort of a puzzle that requires further understanding. One possibility is that there has been a recent description in my previous university, in a study that we did, showing that the subthalamic nucleus actually projects directly to the thalamus. So it could be that because the subthalamic nucleus is a very tremor related nuclei in the Parkinsonian brain, that could be a way whereby the basal ganglia interact with the thalamus and the cerebellar loop to integrate tremor activity. Of course, many of these puzzles and questions that one can pose from the point of view of the classic model are also part of our ignorance and the reduced information that we have. And in the 20-25 years since the model was first put about, there have been many changes. I'm going to summarize few for you. First is this hyperdirect pathway that from the cortex to the subthalamic nucleus that is being histologically and physiologically proven, and in a recent paper published in Movement Disorders Journal by Professor Quartone, you can see here this beautiful tractography showing the fibers arriving into the subthalamic nucleus. Also, the same group have shown projection from the cortex to the GPE, to the GPI, and these beautiful pictures also. I think this is in the March issue. We made it a cover because the figures are really very impressive. This requires, though, a careful histological confirmation. Histologically, it's being confirmed in the mice that the GPE projects back to the cortex. And further work could eventually be proven that there is this loop, cortico-GPE cortex, which could be a new loop and a new way whereby cortex and basal ganglia would be interacting and modulating movement, behavior, and emotions. Interestingly also, the GPE is no longer considered a go-through structure. The GPE projects to the thalamus, as André Perrin showed many years ago, also project to the GPI directly, and back through these archipallial neurons to interneurons in the putamen, and is modulated by these dopamine, extrahiatral dopamine axons to the GPE, GPI, and STN. Therefore, the basal ganglia really is a complex network where parallel loops interact with re-entry loops, subcortical projections, changes internally in the excitability of the nucleus, so that the excitability of these cells actually is very variable and generally highly asynchronous population activity, where firing rate, firing pattern, coupling, uncoupling, oscillatory activity, all that comes together to produce a sort of code of activity. And I'm just showing this just to sort of make you think that we should not be so puzzled. At least I shouldn't be so puzzled after this lecture, because it is so complex. So why should we expect that we were going to understand tremor, rigidity, and akinesia just by one single mechanism? There must be many mechanisms. There must be many ways whereby these different pathways and different functions, different ways of functioning produce symptoms. Anyway, the idea is that, and my prediction is that in the near future, we'll be able to pinpoint a specific circuit and a specific neuronal patterns of activity to understand these major manifestations. I can tell you that with David Marsden, years ago, we were well aware of the complexity of the basal ganglia, and we play through planes, hotels, with these drawings, exchanging these ideas here and there. Here I tell him, need some more inspiration to decide what the GPE does to come. Or here down we could say, Buenos Aires will give me passion to understand, things like that. And this was part of the game. And of course, one of the major interests for Marsden was the paradox, those things that we couldn't understand. The one regarding how palidotomy eliminates dyskinesia is really being very discussed in many events, and I'm not going to deal with this because it's been well published in many occasions. So I'm going to concentrate on this apparent impunity of the basal ganglia to ablation, because after all, if you have any structure and you destroy it, one would expect that there would be some kind of consequence. It just doesn't make any logical sense that one destroyed a structure and all what you get is improvement. What was the function of that structure that has been eliminated? So this notion, we discuss it at length in this paper that continues to be a classic, and I have studied in detail in a number of patients between La Habana in Cuba and Pamplona, where patients who have extremely asymmetrical Parkinson's disease with dopamine depletion in the posterior putamen. We did a lesion in the subthalamic nucleus. The patients had hemichorea, hemiballism, that occurs in 8% exactly of all patients, but in those 8%, the dyskinesia does not go away, does not stop, so we needed to do a palidotomy to eliminate the involuntary movement. So in effect, this patient was heavily dopamine depleted on that hemisphere in the striatum, has the STN block for the indirect circuit, had the direct circuit also blocked because of the palidotomy, and one could say that the basal ganglia in that hemisphere was not working almost at all. Nevertheless, he actually did very well. Okay, I have here cut off part of the window, so I don't see, I have to look at here. So what happened is that he performed very well. The right upper limb mobility improved a lot. He didn't have any problem after we got rid of the dyskinesias with the palidotomy, and careful analysis by Marjan Jashanhasi and John Rothwell in Queen Square, we saw by and large better results on the operated side, except that the patient could not learn, didn't perform well with new implicit learning. In other words, when he has to acquire new information implicitly that was not explicit to him with the affected side, that was wrong, was very, very wrong, was very affected. More recently, Thomas Bureau in Bordeaux has done an experiment in 2 monkeys. Monkeys learn a manual motor task. Then they did bilateral GPI blockade with muscimol in the GPI, and what happened is that the monkey, again like in the patient, the motor learning that had been previously established, they performed well, but they couldn't learn new motor learning. In other words, the same like in the patient, what was already there could be performed, but what wasn't there was defectual. And a very old study from Peggy Anderson and Bob Turner is actually very interesting. They record neuronal activity in the GPI and in the thalamic receiving area, so that they were sure that they were recording in the projection. Then they trained the monkey to do a motor task, and once they has learned, they block the GPI and record and repeat the task with the GPI block. And see, what happened was that the task repeated after blocking, there was no major motor deficit. The monkey did well, performed well, despite having no GPI, and thalamic neuronal activity did not care that the afferent from the GPI was not there. They increased their baseline firing rate, but they continued to respond perfectly well in relation with the task. When the monkey started, stopped, did the sequence, etc. In other words, these thalamic neurons continue to work, to function, to fire exactly as normal, despite the fact that they didn't have the GPI influence coming from the basal ganglia. So altogether, one would say that this experience in patients and monkeys would suggest, just by pure logic, that the basal ganglia is not needed for performing habitual routine learned tasks. That obviously goes tremendously against what my maestro, Marsden, put forward in this magnificent and famous lecture. The basal ganglia deals with the automatic execution of learned motor plans. Now, Marsden was rarely wrong. In fact, he was right almost all the time, even for the wrong reasons. So to think that this is wrong makes me nervous. So I come around to see what is behind this. The essential element of this possible new paradox are performing routine, learned, or automatic tasks, this is all the same, is mediated by the motor circuit, which is the region of the striatum that receives the nigrostriatal projection from the lateral nigra. This is here in the monkey from Hubbard, ventrolateral neurons projecting to the motor putamen in the most caudal part of the motor area of the frontal cortex. The more you go rostral, the more you go into medial substantia nigra and more anterior in the striatum for associative, cognitive, decision-making, and limb emotional components. Performing a habitual or automatic task activate the putamen as predicted, just here, this part of the motor circuit, while when you are in goal-directed, which is when you are not working in automatic mode, when you have to think, when something is new, when you have to take decisions, when everything is not predictable, then the activity in the basal ganglia goes anterior. Therefore, this is the system that deals with automatic movements. Now, in Parkinson's disease, the region which is most vulnerable to and hit by degeneration is the ventrolateral tire of the nigra. This beautiful picture given to me by Professor Halliday shows this wonderful cell loss here compared with this normal brain of a patient, showing exactly where these cells are lost, giving rise to this dopamine loss in the posterior putamen and the reduce and the impairment of automatic movement. So all these fit. If you lose dopamine in the sensory motor region of the nigrostriatal system, automatic habitual tasks are first impaired because this is what is most, this is what they take care about. To lose neurons there has to be a number of vulnerability mechanisms. And for instance, it's been shown that one single axon in the rat arriving to the striatum arborizes and makes something like 200,000 synapses for the sensoromotor striatum, while in the limbic striatum it does an order of magnitude less, something like 20,000 synapses. So it's been hypothesized that all these non-stressors like mitochondrial, calcium influx, protein, oxidative stress, etc., may be acting through a system which is under stress because of high energetic consume, as put by Paul Bolam in this paper in Movement Disorder. If you are under such energy stress, many of these stressors will be harmful and will impair the physiology of the system and perhaps cause the degeneration. But the problem here is that if performance of an acquired action doesn't rely anymore on basal ganglia output, and therefore the motor basal ganglia circuit is not activated, why is then that these neurons are going to be overactivated if we don't need them? So I see here a major puzzle and paradox. So how am I going to be out of this? Classical studies by Wolfgang Schulz in the 90s have shown that dopamine cells in the nigra only fire, as shown here, in response to reward and not to movement. And that, of course, supported this idea. But in fact, this has changed radically recently. And here is a study in the monkey by Matsumoto indicating that VTA cells do respond to reward or don't respond, do the opposite, silence with a noxious stimulus like an air puff, while the lateral dopamine neurons in the nigra respond equally to either stimulus. They work this out, and they show that the lateral substantia nigra actually show responses to a number of situations, relations with motivation, silencing, orienting, cognitive processing, etc. And this could be a sort of the dorsolateral, not the ventrolateral, but the dorsolateral. But this, to begin with, modify the idea that substantia nigra cells don't fire in relation with activities in the external world. Therefore, one would have to conclude that they are more frequently activated by different stimulus and tasks. Here, now I have a problem here to find the, okay. This is a study, very important study from Rui Costa in Lisbon. The mice learns to do a sequence of activity going from one point to the other to find the reward. The reward is given randomly, so he cannot move according with the reward. He has to learn the sequences, and once he learns the sequences, he will continue to do that regardless when the reward will be given, which is the experimental definition of a habitual task. So those mice learned the task, and then it was shown that neuronals in the substantia nigra fire physically in relation with the beginning or the end of each of these sequences of movement, and they were here in the lateral substantia nigra, and also neurons in the striatum also fire at the beginning and at the end of the sequence, and these were neurons mainly in the sensory motor striatum. Therefore, self-initiated habits elicit phasic dopamine responses. More so, and recently, in this study, have shown that, in fact, the mice shows a kinematic direct relationship between acceleration of the head and spiking activity in the nigra, so that a very straight, highly significant line could be built between the movements, the acceleration, and the spiking, either for the cue to move or the actual reward. And the authors claim that they have therefore disengaged the reward part of the self-response with the actual kinematics of the response. So one could have here a situation whereby this new cortico-nigra projection can modulate neuronal activity of the nigra. We have our all well-known direct cortico-subthalamic nigra projection also, and the substantia nigra is no longer isolated. The sensory motor substantia nigra is no longer isolated from the basal ganglia and is actually overactivated probably most of the time. Therefore, and this is schematic, speculative, imaginative, when we do a routine motor task, we are stimulating our motor circuit and our lateral substantia nigra neurons. When we do something more cognitive, associative, this is the circuit, and here we stimulate both. We recruit the more medial neurons, but these also continue to fire when the situation is emotional, then the accumbens and the limbic, but also the motor part of the nigra. In other words, whichever we do, according with this proposal, these cells in the nigra ventrolateral, the ones who die in Parkinson's disease first, are activated and recruited. Even more, it is now known that we learn from goal-directed to habit formation, and that is this switching another important task of the basal ganglia and the nigrostriatal system. But it's also the case that we have a system for inhibition, and we have goal-directed inhibition and habitual inhibition, which probably activate the same corticobasal ganglia circuit as the moving part of goal-directed and habitual activity. So this is just totally imaginative, but I would have thought that it's highly likely that dopamine cells are also engaged in the formation and in the monitoring of habit in terms of inhibition, not just habit in terms of action, giving another loading to the system. So a wide range of events activate sensory motor dopamine neurons. They correlate with movement kinetics. Automatic habitual tasks engage sensory motor dopamine response, and with Ledia Hernandez and Pete Redgrave, we're doing studies now in the rodents to show that indeed all these features are risk factor for cell vulnerability in Parkinson's disease. Indeed, I think these cells, the ventrolateral nigra cells projecting to the motor putamens, die because they are over-engaged. And my prediction is that the more things we do in our new world, the more they will engage and the more vulnerable they will be. So just to wrap up, there are several points which remain to be discussed. Why do patients tolerate lesion of the basal ganglia so well? I can say that they tolerate it, but not that well. If we look carefully, they do have errors, like inhibitory errors, defectual errors to do simultaneous activities, etc. So this is not completely crystal clear. Primates in those experiments have been overtrained to do one thing, and there is nothing else they have to do when doing the experiment. Therefore, the need to use the basal ganglia in those circumstances is low, and therefore this could explain why they perform much better. More worrying to me as a clinician is that while one could understand and concede that akinesia is the pathological counterpart of movement control, what is the physiological meaning, if you wish, of rigidity and tremor? What is it? What would it be? What is rigidity telling us in terms of motor control? What is tremor? Is tremor just a noise, or is revealing some kind of physiological mechanism? That has to be understood. And coming to my primary interest, if multitasking leads to functional overload of the nigrostriatal system, how can the focal asymmetrical motor features of PD at onset be explained? This leads to intriguing daily life features, such as the risk of Parkinson's disease will be influenced by features such as predominant body part use, for instance, hairdressers versus professional cyclists, or task versus multitask, dependent occupation, an assembly worker versus a designer, or differences between people who are focused and do one thing at a time versus those who are constantly engaged in several activities simultaneously. Those are questions that may be out there and that one needs to try to find out, and these are just ideas. So I finished my lecture with a proposal that we put forward that the nigrostriatal projection, and in particular the sensory motor region, is engaged in the control of learned habitual behavior, a demanding function during simultaneous and multi-domain action. This, in turn, is a major vulnerability factor for the ventrolateral neurons of the substantia nigra ventrolateral region in Parkinson's disease. This is my new center in Madrid. I'm part of the group, growing as it is. Ines and Raul have worked a lot with this PowerPoint, and Ines is making experiments in the monkey to prove some of these ideas. Pete Redgrave in Sheffield is like my guru, and Maria Rodriguez-Oroz, my former colleague, continues to help. And I can't think any better way to finish than by reiterating this writing that David Marsden wrote, because those papers that I wrote with him, of course, he was the one who knew English, as you can easily understand. So we can say that no doubt our difficulties will be explained by future knowledge and our speculations will be disproved. But we hope they will spur others to make the discoveries that will explain the paradoxes we find so difficult and that will disprove our theories put forward here to tell us really what the basal ganglia do, to which I like to add and why neurons die in Parkinson's disease. Thank you very much.
So that concludes the session on the presidential lectures. I want to remind you that at 10:30, there's a very special plenary, 25 years of movement disorders grand rounds. This is a can't-miss opportunity. So please come back in a half an hour and we'll see you there. Thank you very much.